Hi, my name is Veronica Taylor, and I am a graduate student in the biophysics program at University of Michigan. To start this video off, let's talk about you. You seem like a single entity, but you're actually a sum of parts. You're made up of things like muscle and tissue, and these are in turn made up of many cells that, in turn, enclose many tiny proteins. Even though you probably think of your body as a fairly stable object, there are all these dynamic, microscopic events constantly occurring inside you. How do we know this? Scientists in the field of structural biology can use powerful techniques to visualize and model proteins at a level that allows us to see their atomic structure. To demonstrate an example of all this, today I'm going to tell you about some protein-protein interactions and dynamics in a pair of proteins involved in blood pressure regulation. By studying these interactions using a technique called nuclear magnetic resonance, I am hoping to gain a greater understanding of heart-related diseases, which could lead to development of better drugs for treatment of these diseases. But let's back up first. Proteins, you might be asking yourself? Like the protein in my food? Well, technically yes. All animals and plants are made up of these microscopic entities we call proteins. And when we humans consume them from a food source, we break these proteins down into their still smaller subunits called amino acids. Amino acids are the building blocks of proteins. When the individual amino acids are strung back together into longer strings, our body can make new proteins that we humans need to function. There are millions of different proteins known, and they each have distinct structures and properties. Here are two different proteins. The one I've colored in green on the left produces a glowing green color in jellyfish, and the one I've colored in red on the right helps our eyes sense light. Note the difference in how these proteins look. This one has long, flat areas that we call beta sheets in its structure, and this one has a loopier look because it has what we call alpha helices in its structure. Maybe you've heard the architectural phrase, form follows function? This idea holds true in protein structure as well. The shape of the protein is closely linked to its functional role in your body. I'd also like to emphasize that even though these structures look frozen in place on paper, in reality they are constantly in motion. Some parts are just wiggling around in place a little bit, and so aren't as interesting, but some parts can move a lot, swinging about or even rotating around the protein. These are some of the most interesting parts because they are usually helping the protein perform its job. Sometimes multiple proteins can physically come together and interact to cause changes in each other's structure. For example, when you open the lid of a garbage can by pressing the lever with your foot, your toes interact with the lever. This physical interaction causes a change in the structure of the garbage can, the opening of the lid. Proteins can also produce structural changes in other proteins by physically coming in contact with them. Studying these movements can help us learn how proteins interact and keep our bodies working. How do we study these movements? Scientists have many techniques available, but I'd like to talk to you about one called nuclear magnetic resonance that I use to study a protein involved in blood pressure regulation. I'm going to tell you about this protein, and then I'll get to how I use this technique. Here is the protein p63-ROGEF. I'll call it p63 for short. Scientists have found that patients with high blood pressure also have higher levels of p63 when compared to patients with normal blood pressure. This suggests that too much p63 could possibly be a cause of high blood pressure. p63 interacts with another protein that is well known to affect blood vessel and heart functions. This other protein is called G-alpha-Q, or GQ for short. The functioning of p63 inside the body is not always constant. It must be turned on first when the body needs it to ultimately help constrict blood vessels. GQ is able to turn p63's function on by physically interacting with it and changing p63's structure somehow to put it into an on state. When GQ leaves p63, thus ending their interaction, p63 returns to its off state. Scientists have so far determined the structure of p63 and GQ when they are interacting, shown here. Here's p63 on the left, and here's GQ on the right. you can see they are very close in the region between them, which is their interaction surface. I would like to describe any structural movements that occur in P63 when it interacts with GQ. I'm hoping that by studying the movements of P63, we can then develop a drug to target P63 and possibly treat high blood pressure. The picture we have is a great starting point, but there's still more work to do. Imagine you have a picture of a person suspended in midair. Now tell me, are they jumping up or are they falling down? You can't be sure unless I give you a picture taken a moment before. If the before picture shows them on the ground, then they are jumping up. And if the picture before shows them even higher in the air, then they are falling down. But without that picture, you can't say which is the case. This is the same problem concerning how P63 is turned on. We have an after picture of GQ and P63 interacting, so we know how P63 looks when it's turned on. But we don't have a picture of P63 before GQ decided to interact with it. What does off look like? Knowing the full process will aid in the development of a drug to target P63 and treat high blood pressure. I am using a technique called nuclear magnetic resonance, or NMR for short, to determine the three-dimensional structure of the off version of P63. NMR is a technique for determining protein structure and the dynamical character of its structure using a large magnet. I use electromagnetic energy to cause changes in certain properties of the atoms that make up the amino acids of my protein while it is inside this magnet. I can then detect the resulting electromagnetic radiation that is given off by the protein sample using receivers connected to a computer. The change that each atom undergoes during the experiment is dependent on the environment in which that atom is located in the structure of the protein. Atoms near each other will experience similar things in their environment, and so the data I collect for them should reflect this. To illustrate this idea, pretend you have a group of 30 people huddled together. You have a list of their names, but without looking at them, your goal is to draw a map of where they're all standing in the group. If group member Fred says he's standing next to Mary and Paul, and group member Bill says he's standing next to Paul and Jane, then you could surmise that Paul is standing between Bill and Fred. NMR data works in a somewhat similar way, where the people are atoms, and this is how scientists piece the data together to eventually yield a three-dimensional model of the protein and also model how the protein moves. Here I have pictured real data from an NMR experiment. Each amino acid in the protein has a nitrogen and hydrogen bonded together, and each of those bonds can be represented by the dots shown here. In a way, we can say that each dot represents one amino acid in the protein. The location of the dots in the spectrum depend on the environment of the nitrogen and hydrogen in the bond. We use multiple types of experiments that give us data looking similar to this one to piece together which dots are which amino acids and where those amino acids are spatially in the protein. Once I have a clear picture of how P63 looks in its off and on states, scientists can apply drug design strategies to aid in creating a drug to target P63. This drug might either prevent GQ from interacting with P63, or it could lock P63 in its off state. One such strategy is called structure-based drug design. To illustrate this strategy, let's take a minute to imagine you're buying a shirt as a gift. If you know whom the gift is for, then you are more likely to buy an appropriate shirt. For example, the right size, or maybe a color that fits their wardrobe. But if you don't know whom it's for, then all bets are off. That shirt is likely just to be returned to the store later. This is how structure-based drug design works. Design the drug based on what you know about the protein's structure. This requires that you already know a fair bit about the protein in question. The aim of my research is to obtain the structural knowledge needed so that a drug can be designed to target P63. So to wrap up, by using the technique of NMR, I can obtain a three-dimensional model that is a snapshot of the off state of P63. I can then compare it to the model we already have of P63 in the on position. This should give a clear enough sequence of events that will allow me to fill in what happened between the before and after photos. Structure-based drug design can then be used, and who knows, years from now, everyone might be taking the latest blockbuster drug for high blood pressure targeting P63.